Okay, students, today we will discuss a new topic. That is the last topic, I think, in our syllabus, that is antifungal agents. Antifungal agents are those drugs which can be used against fungal infections. And normally we have discussed about antiviral agents. Then I have mentioned certain challenges of antiviral drugs because viruses live inside the host cell, and it's difficult to kill the viruses only without affecting the host cell. That is an important challenge of antiviral therapy. Okay. Whereas the antifungal agents has also got certain negatives, because fungus which affects human beings, which causing fungal infections, and they are usually eukaryotic organism, and our host cell is also eukaryotic organism. So anything which inhibit the eukaryotic cell itself will inhibit or itself will kill our host cells also. That is an important challenge of antifungal agents differing from the bacterial cell. That is important. So just like antiviral agents, antifungal agents has also got certain negatives, and so we have to find out a suitable antifungal candidate by targeting specific regions or specific sites in fungi which is not present in eukaryotic host cell. So that should be identified, and that should be considered as the targets of antifungal agents. I think the idea is clear for you, because this the target of antifungal agents should be different from the host cell. That is the thing. So otherwise it will kill the our host cell, eukaryotic cell also. So that targets include, there are three different type of targets are the one is actually the cell wall. The cell wall of fungi, we don't have a cell wall, so but fungi have a cell wall. So and that cell wall is made up of chitin. So bacterial cell wall is peptidoglycan, but the fungal cell wall is chitin. So we can inhibit or can the growth of fungi by inhibiting the formation of chitin cell wall. Okay, chitin is biochemically, it is N-acetyl glucosamine polymer of N-acetyl glucosamine. And also we can target the plasma membrane, because our plasma membrane and the fungal plasma membrane, they differ in the feature that our cell membrane contains cholesterol, the sterol form as cholesterol, but the fungi, they contain the another sterol form which is known as ergosterol. Human cell membrane is composed of cholesterol, but the fungal cell membrane is formed of ergosterol. So that also is a target for antifungal agents. And then the antifungal agents, the nucleic acid synthesis, the DNA polymerase enzyme and all, that can also be considered as a target, because DNA synthesis of fungus eukaryotic promoters are different. Okay, that is important. Eukaryotic DNA polymerization is that's different. So these are the important targets of antifungal agents. So we can target at chitin synthesis or the ergosterol formation or the nucleic acid synthesis. These are the different targets of antifungal agents. And the antifungal agents, we can broadly classify it into two types: the systemic antifungal agents and the topical antifungal agents. Systemic antifungal agents are those agents which are used for the treatment of systemic mycosis. Systemic mycosis. Whereas topical antifungal agents are those which actually act upon the superficial mycosis. Superficial mycosis, not a deep infection. So that is the thing. So first of all, we will discuss about systemic antifungal agents. There are actually different types of systemic antifungal agents. We will discuss one by one. The first type of systemic antifungal agent is polyene antibiotics. the polyene antibiotics, the first type of uh, uh, systemic antifungal agents, because it is called as antibiotic because it is produced from actinomycetes, but it does not have an antibacterial activity. It will be having an antifungal activity. And what is the mechanism of this polyene antibiotic? They bind to ergosterol in the fungal plasma membrane, leading to the formation of pores and hence increased permeability of the cell membrane. This allows the leakage of intracellular ions and enzymes, especially the loss of intracellular potassium, causing the death of the fungus. So how it acts? It is acting on ergosterol. It will bind to ergosterol and produce pores, and a leakage of materials, especially potassium ion, lead to the death. They bind selectively to ergosterol in the fungus but not the cholesterol in mammalian plasma membrane. Those are important of this polyene antibiotics. They will bind only to the ergosterol, not to the cholesterol. But so it will kill only the fungal cell, not the animal cell, okay, or the host cell. And we'll see one example of a polyene antibiotic, which is known as amphotericin B. Amphotericin B is the most effective antifungal agent used for systemic mycosis. And it is actually a fungicidal antibiotic without an antibacterial activity, and it is derived from the species Streptomyces nodosus. Streptomyces nodosus. And how it acts? The amphotericin B interact hydrophobically with ergosterol in the plasma membrane. You can see it will bind to the ergosterol and that will form a pore, and through this pore potassium and other small molecules are lost through this pore and causing the death of the cell, and that lead to cell lysis. Also, this amphotericin B stimulates the release of cytokines from the mammalian phagocytic cell, which also improves its antifungal agents. So, it stimulates the release of cytokines. And this amphotericin B has got a broad spectrum of activity against different type of systemic infections like candidiasis, cryptococcosis, histoplasmosis, etc. So it is active against different type of systemic mycotic infection. So this is an example of a polyene antibiotic. Now we'll see the second type of systemic antifungal agent, which is known as broadly as heterocyclic benzofurans antibiotic. So it's actually its classification is based on its chemical structure. So heterocyclic benzofuran, it has got a furan ring and it's heterocyclic, and it's also an antibiotic which is obtained from penicillium. Okay, penicillium, sorry, some of them are penicillium, but one example is obtained from penicillium. And one example of this antibiotic is griseofulvin. So griseofulvin is an example of a heterocyclic benzofuran antibiotic. It is actually a narrow spectrum antifungal agent and isolated from Penicillium griseofulvum. It is obtained from Penicillium griseofulvum species, and they act as actually inhibitor of mitosis. So it is acting as a fungistatic agent. You know what is fungistatic? It will inhibit the growth of the fungi. It will not kill the fungus, whereas the amphotericin B that will kill the fungal cell. So it is actually a fungicidal antibiotic, but this is fungistatic drug because it will suppress, or it will inhibit the growth of the or multiplication of the fungi. And what is the mechanism of action? It actually inhibit mitosis It inhibits fungal mitosis by inhibiting the mitotic spindle formation. So it's know the different steps of mitosis and also the importance of spindle fiber formation for the movement of chromosomes to opposite pole. That is very important. So it is actually inhibiting mitotic spindle formation. As a result of this, fungal cell will not be killed, but it cannot replicate, or it cannot multiply or divide. The drug actually binds to microtubule. Microtubule are very important in spindle fiber formation and interfere with this microtubule formation, and thus inhibiting mitosis by preventing the formation of mitotic spindle formation. I have explained you one example. This is griseofulvin, which is obtained from Penicillium griseofulvum. So this is the second type of systemic antifungal agent, that is heterocyclic benzofurans antibiotics. Now, the third category of systemic antifungal agents are azoles. Azole drugs. Azole drugs are very common antifungal agents. We usually take azole drugs for a fungal infection. It's the most widely used antifungal agent, and we can classify the azole drugs into two broad categories, that is imidazoles and triazoles. This classification is based on the chemical structure. That means it is actually based on the number of nitrogen atoms in the azole ring. The azole ring, organic ring formation, the azole ring, the number of nitrogen, it will form imidazole and triazoles. And there are different examples of imidazoles and triazole. Different imidazole rings include ketoconazole, miconazole, clotrimazole, etc. Examples of triazoles include fluconazole, itraconazole, voriconazole, etc. So there is no need to study about each antifungal agent in detail. Just we are giving a brief discussion, but we will see the mechanism of action of azole drugs. They are also fungistatic agents. You know what is fungistatic agent? Actually, how they work is that they inhibit the cytochrome P450 dimethylase enzyme. They actually inhibit one particular enzyme, cytochrome P450 dimethylase enzyme, which is very important in the formation of ergosterol. And as a result of this inhibition, the ergosterol formation will not occurring, and this inhibition disrupt the membrane structure formation. You know the importance of ergosterol in fungal cell membrane, and also its function will be affected, and that inhibits the fungal growth, and they are usually fungistatic agent. There are actually different types of normal. When you have a particular fungal infection, we take no, different types of azole, that is ketoconazole, fluconazole, etc. So, and also similar to its mechanism of the action, usually the fungi that will develop resistant to azoles by mutation in the gene coding for demethylase enzyme gene mutation will occur, and the fungi will become, they will survive and they will become mutant. That is another important feature. Okay, we'll discuss the other things in the next session. Thank you.